on as we start, but we'll get going. Okay, good morning, and welcome to this month's GCE webinar. As we move into our third year of this webinar series, we hope to expand the breadth of speaker backgrounds to not just include established GCE scientists, but also the up-and-coming generation of senior graduate students and postdocs who are looking to take their careers to the next stage and gain exposure in the field. In addition, the GCE community is becoming increasingly aware that there's a lot more to GCE than just tRNA synthetases and tRNAs, and that we as a field probably need to do a better job integrating with those fields of, say, ribosome chemistry, structure, and function, and better understand how these translational machineries can or can't tolerate non-canonical amino acids into growing peptide chains. And for that reason, this year, we're really excited to bring in more scientists who aren't necessarily GCE developers per se, but also those in the fields of protein translation biology. And so we hope our two speakers today, Dr. David Bedwell and Philip Ilievsky, help serve these two important goals. First up is my distinct pleasure to introduce Dr. David Bedwell, professor and chair of the Department of Biochemistry and Molecular Genetics at the University of Alabama at Birmingham, where he has built a distinguished career over the last 36 years. Dr. Bedwell earned his BS in microbiology from Purdue University in 1979, followed by a PhD in molecular biology from the University of Wisconsin-Madison in 1985. He completed his postdoctoral training at Caltech in 1988, where he trained in the molecular mechanisms and regulation of protein translation. His research interests center on developing therapies for diseases caused by premature translation termination codons, or PTCs, which are responsible for a variety of genetic disorders such as cystic fibrosis. Among his notable contributions, Dr. Bedwell is known as the pioneer in developing nonsense suppression therapies. His lab conducted the very first study that demonstrated suppression of premature translation termination codons could restore protein function in CFTR, a groundbreaking achievement published in Nature Medicine in 1996 that has since shaped the course of cystic fibrosis research and therapeutic strategies. His lab has also identified compounds capable of improving the efficacy of these treatments and is expanding these approaches to other diseases such as Hurler syndrome and Rett syndrome. Dr. Bedwell's work has been widely recognized with many accolades, including the prestigious James C. and Elizabeth T. Lee Endowed Chair of Biochemistry and election as Fellow of the American Academy of Microbiology. Dr. Bedwell, it is an honor to have you here today. Please take it away.
All right. Thanks a lot, Rick. All right. So today I'm going to talk about translation termination and mechanisms to inhibit the process of translation termination. I don't know hardly anything about genetic code expansion, but I think I might have a few tidbits that might be useful for your community. So first of all, let me get there you go. So here's my disclosures, and let's start off by just talking about the mechanism of translation termination in eukaryotes. So over here on the left, we have a pre-termination complex where we have a peptidyl tRNA in the ribosomal P site, a stop codon in the ribosomal A site, and a ternary complex containing the translation termination factors ERF1, ERF3, and GTP will bind to the ribosomal A site. ERF3 is a GTPase, so its hydrolysis of GTP will facilitate a conformational change that leads to a combination of ERF1. ERF1 is shaped like a tRNA, actually. So one end recognizes the stop codon and the other end is up near the peptidyltransferase center of the ribosome, or it can facilitate hydrolysis of the ester bond between the peptidyl tRNA and the completed polypeptide chain. This accommodation only occurs, though, after certain proofreading steps have been done to ensure that the stop codon is, the codon in the A site is really a stop codon. Following peptide release, we then are left with a post-termination complex that must be disassembled and recycled. All right, so here's the stuff for my nonsense suppression studies. In-frame nonsense codons actually introduce a premature termination codon, which I'll refer to as a PTC in the mRNA. These account for about 11.5% of all known mutations that have been shown to cause inherited genetic diseases. So these are quite common. And if you think of just about any genetic disease caused by a loss of protein function, there's going to be a subset of 5 to 10% that are caused by nonsense mutations. All right. And second, approaches to suppress nonsense mutations could provide potentially a therapeutic benefit for many patients with PTCs. All right. So here's how it works. So we have a gene in the nucleus, which gets transcribed, processed, cap, spliced, polyadenylated, and it gets exported out into the cytoplasm, where it undergoes what's referred to as the pioneer round of translation. There are two consequences of this if you have an in-frame nonsense mutation designated by the red stop sign with an S. First, you get premature termination, which results in a truncated polypeptide, which is normally non-functional and unstable. And second, this leads to a process called nonsense-mediated mRNA decay, or NMD, which reduces the abundance of the mRNA itself. So if you look at what's happening at the ribosomal A site, normally termination is very efficient, let's say 99.9%. However, there is the possibility of a near cognate aminoacyl tRNA binding and incorporating its amino acid at the stop codon. And this may occur at roughly about 0.1% normally. So this is an equilibrium, and what we want to do is push this equilibrium away from termination and toward near cognate incorporation. All right. So the way we do that is with drug-induced read-through, and let's say we get 1 to 10% read-through instead of 0.1. That can lead to 10 to 100 times more of the full-length functional protein, and this can be mediated with classical compounds called aminoglycosides or adelurin, which has been around for quite some time. But I'll talk today about some new classes of compounds that we think are going to be more useful. All right, so what about this nonsense-mediated mRNA decay thing? There's two flavors of this. One is called core NMD, or sometimes long 3' UTR NMD. This can occur without exon junction complexes. And basically, what happens is when the termination complex recognizes a stop codon on the ribosomal A site, an NMD complex can form here under certain conditions. For example, when there's a long 3' UTR, and this can lead to recruitment of not only the NMD machinery, but also subsequently RNA turnover factors. This is common evolutionarily from yeast all the way to humans. It's pretty low magnitude, 2 to 4 fold is all. The second form is EJC-dependent NMD, and this is a much more powerful 10 to 20-fold type effect on mRNA abundance. And in this case, you assemble what's called the SURF complex at the stop codon with the release factors SMUG1 and UPF1, and they look downstream to look for the occurrence of an exon junction complex, or EJC. This is deposited on the mRNA during splicing, and normally stop codons are in the last exon, so there shouldn't be an exon junction complex downstream. And when it does detect this, that leads to recruitment of additional factors and degradation of the mRNA. Okay, so the next question is, should we worry about this process? So, well, if you're getting a reduction in mRNA to 10% of normal, if you got 100% read-through on that mRNA, you'd still only get 10% of the wild-type level of protein. So this can be a serious problem. However, we've done some studies with luciferase-based reporters, such as this one shown here with a nanolook with a PTC upstream, followed by a globin gene with introns. So this actually is responsive to PTC read-through as well as NMD. So we call these read-through / NMD reporters. So we can take this configuration here and treat it with various nonsense suppression agents, singly and in various combinations, and we can get increasing levels of read-through that you see plotted here going up to almost 800-fold above background. And what we found when we did qPCR was the more read-through we got, the more we stabilize the mRNA. And so the way we interpret this is that it's something like this. So on a normal termination codon, where you have the ribosomes transiting down the entire open reading frame, it displaces various RNA binding factors such as NMD factors. And so they have little effect on the stability of the mRNA. The only ones you have remaining are those in the 3' UTR. However, when a PTC occurs, the stop codon terminates translation early, and these things are able to bind and recruit NMD factors and mRNA turnover factors. So you get a reduction in mRNA abundance. So what we think is going on over here is that the more the ribosomes are plowing through and disrupting these factors binding to mRNA, the more we stabilize the mRNA. So the idea here is if we get enough read-through, NMD is not going to be a problem, okay? All right. So what about these various mechanisms? So we've learned a lot in the last few years. Here's a ribosome with a peptidotRNA in the P site, and here's the competition between the near cognate tRNA and the ERF1, ERF3 complex. So we have various factors that are small molecules that can push this equilibrium toward tRNA incorporation. One is a class of compounds called ERF1 degraders. These are small molecules that are molecular glues that degrade ERF1. There are ERF3 degraders that are also molecular glues that get rid of ERF3. There are aminoglycosides that bind to helix 44 of the 18S ribosomal RNA, which is right at the base of the ribosomal A site. And these compounds influence the fidelity of proofreading of the incoming aminoacyl tRNA and allows more incorporation of these near cognates. And then we have another class that enhance the activity of the aminoglycosides. And then, of course, there's adalurin, which has been around for a long time. It's a very safe compound, but it's pretty low efficacies, which is why so far it hasn't been able to become approved for clinical use. All right, so I'm going to tell you a little story next about how we identified some of these classes of compound, one in particular, the ERF1 degraders. So we worked with Southern Research, which is a contract research organization at down the street from us in a project funded by the CF Foundation. Did a high throughput screen of over 750,000 compounds. And Southern Research has the high throughput screening capacity as well as a chemistry group that is very good. So we found, as you can see, many compounds that seem to induce read-through. And I'm going to tell you a little bit about one called SRI 37240. So here's our initial screening reporter. We're getting a nice response with a good EC50 of 6 or 7 micromolar. We have various flavors of our read-through reporters. This one shows read-through only, and it's going up a lot less than the dual read-through NMD. There is some effect on NMD, probably just because of read-through, as I showed you on the previous slide. And then we always test these with a wild-type control just to make sure we don't get something that enhances transcription initiation or something to make, you know, more of the luciferase reporter. All right. So one of the first things we always get asked when we identify nonsense suppression agents is what happens to normal stop codons. So we've done ribosome profiling here, which is kind of like RNA-seq, except you purify polysomes, RNase treat, so that the only thing left is the mRNA bound by a ribosome, and then we subject that to sequencing. Instead of getting a uniform distribution of reads, we get these things with intervals of 3 on open reading frames because the ribosomes translocate codon by codon, and it's intervals of 3 nucleotides. So here's the herringbone feature in the upstream open reading frame. This is an alignment of all the reads in a cell. So these are natural stop codons. And you can see with the DMSO control, we get a pause at stop codons. This is always seen. This indicates that termination is less efficient than the elongation process. When we add G418, which reduces fidelity, the pause is shorter, which suggests it's, you know, clearing the stop codon faster. And with our 37240, the pause is prolonged, suggesting the stop codon the drug makes the ribosome sit on the stop codon longer. If we take this region just downstream of the stop codon and blow this up, what we see is DMSO gives us very little read-through. G418 does give us read-through, but our 37240 superimposes on the DMSO, indicating that this ERF1 degrader does not mediate read-through of normal stop codons. Okay. All right, so then the chemistry group got involved. We liked this one. And so they made a number of derivatives and came up with 41315, which is shown here. And here is the original 37240. And here is 41315 alone. So you can see it's much better as far as efficacy. And then we also always do minus and plus G418 because we can show synergy with G418. And you can see there's a big synergy here. This one, the synergy is not as high, but it's still going higher than the original 37240. So this is the molecule that we've now gone forward with. The way this works, it turns out, is it acts by leading to the degradation of ERF1. Here's an ERF1 decay curve, and it has a DC50 of about 1.6 micromolar. And basically here is normal cells, extract ERF3, ERF1. Here is after treatment with 41315, and you see ERF1 is completely gone. And then when we add a proteasome inhibitor, MG132, we can block that degradation, okay? So that suggests that ERF1 degradation is proteasomal dependent. All right, so if we can block degradation, we should be able to inhibit translational read-through as well. And so we tested that here with our reporter assay. It turns out that translation itself is very sensitive to proteasome inhibition. So it's very easy to block translation with a proteasome inhibitor. So we had to use very low levels. But here you can see with the conditions we use, we could show a significant decrease in read-through under the conditions where we got rid of ERF1 degradation. This is with our reporter, and this is with CFTR function itself. CFTR is a cyclic AMP activated chloride channel that's defective in the disease cystic fibrosis. All right, so to give you a little preview of how these things work. So ERF1 degraders have been shown to be a molecular glue that actually glues ERF1 into the ribosomal A site. That leads to a block in translation termination. This induces a ribosome quality control mechanism, which leads to the recruitment of two ubiquitin E3 ligases, RNF14 and 25. They come in and ubiquitinate ERF1, and it then goes to the proteasome for degradation. Consequently, as ERF1 is degraded, two other things are happening. First, we induce the integrative stress response, which is mediated by an empty A site, or a blocked A site, or an empty A site. GCN1 and GCN2 are recruited and leading to EIF1-2 alpha, EIF2 alpha phosphorylation, and translational repression. That's to give the cells a chance to clean up this mess and get things working again before translation resumes. And then a more severe effect is ribotoxic stress response, or RSR, where ZAC alpha recognizes this. So what happens is the first one pauses, the second ribosome runs into it, causing a collision. ZAC alpha recognizes this collision, leads to phosphorylation of P38 and junk, and that can lead to a whole cascade of things that are generally not good for the cell. All right, so that's the ERF1 degraders. The ERF3 degraders are completely different mechanism, and it's a simpler mechanism. So you have an E3 ligase complex called cereblon, which normally does not recognize ERF3, but these small molecules act as molecular glues that will glue the small molecule to this cereblon complex, leading to its ubiquitination and degradation. All right. All right, so going back to the ERF1 degraders, 41315 treatment, well, we wanted to look and see how these things were working. And so when we did RNA-seq, we found the induction after 41315 treatment of a series of genes, including GAD34, ATF3, CHOP, and ATF4. These are all key components of the integrative stress response. And so what we have here is a cartoon of what happens when we induce ribosome collisions. GCN1 and 2 bind to the collided ribosome, leading to the phosphorylation of EAF2 alpha and induction of the integrative stress response. That's with relatively low stress. And as stress gets more severe, you can induce the ribotoxic stress response, as indicated here by the needles in the red, and that leads to P38 phosphorylation, junk phosphorylation, and potentially apoptosis. All right. So ISR is induced, and here is RSR. So we did these series of experiments with the ZAC alpha plus and ZAC alpha knockout line that we got from Ron Weck at Indiana University Medical School. Here is the ZAC alpha. There's a phantom band there, but clearly this is a good knockout of ZAC alpha. Here's the ERF1 going away when we add 4, 1, 3, and 5. P38 is induced with the wild type, but is not induced with the ZAC alpha knockout, indicating that ZAC alpha is required to induce P38 phosphorylation. And then here's junk phosphorylation. Here you see very significant junk phosphorylation, but not with the ZAC alpha knockout. So take-home message here is this is ZAC alpha dependent, indicating that it's mediated by the ribotoxic stress response. So we were concerned about this junk phosphorylation in particular. So we monitored this with a time course. You can see phosphojunk going up and up and up to 12 hours after addition, and then it goes away. So it seems to be self-limiting here. And we're still trying to figure out exactly how this is working. We also tested for cell viability with a Cas-based Glow 3.7 kit from Promega, and basically we saw no decrease in cell viability. Here's the bortezomib positive control. So the conclusion here is that this attenuation appears to prevent apoptosis from happening, and the cell viability is not reduced. All right, so the last little tidbit, we recently published some work with Alan Jacobson's lab. They had made or used machine learning to make an algorithm that would predict the susceptibility of stop codons for read-through in eukaryotic cells. And so they approached us about making a bunch of CFTR specific reporters with CFTR known nonsense mutations. And so we made our reporters. So their algorithms based on about 20 different parameters, long upstream regions, downstream regions, the identity of the stop codon, the first base after the stop in particular, the length of the 3' UTR, all these different parameters. We made our reporters exactly the same except for the 3 upstream codons, the identity of the stop codon, the 3 downstream codons. And what we found with our reporters is we had a 70% correlation with their algorithm, indicating that these parameters are sufficient to predict the susceptibility of a stop codon in its local sequence context. This is, I think, important for your work because genetic code expansion, you need to make sure if you want to get good read-through, you want to have a genetic, a nonsense mutation in the optimal context. But for our purposes, this is a very interesting result because there's only been one clinical trial. Analurin was tested, and although some patients responded well, many didn't. Remember, I told you this is a pretty weak read-through agent. And basically what we predict is that if we now use this algorithm in upcoming clinical trials, we would be able to cherry pick the most responsive patients and then actually increase our chances of getting approval for a clinical trial. All right, so the take-home message here. Translation community has identified a number of read-through compounds that suppress PTCs to significant extents. We're now understanding how these the mechanism of these compounds, which should aid further accelerate the path of future clinical uses. These can be suppressed by classes of read-through agents. These PTCs can be suppressed by certain of these agents where normal termination codons are not affected, which is critical. We've identified an algorithm to predict the relative susceptibility of a PTC for read-through, which should be useful for clinical trials in the future. And for your community, the sequence context of nonsense codons should be considered to ensure optimal suppression. All right, and those are the people that did the work, and I'm happy to take questions.
Thank you, Dr. Bedwell. Really fantastic talk and super exciting science. We'll open the floor to anyone who has questions. Ryan?
David, thank you so much. And one quick question relating to the relationship of the suppression. Obviously, everything you're doing in a eukaryotic cell is being suppressed. It seems like it's being suppressed by near cognate suppression. First, do you know what amino acid or tRNA is dominating that near cognate suppression? And I guess B, in a follow-up, since what we're doing in GCE is obviously putting in an actual, stop codon, do you think that that your approach would increase near cognate suppression in GCE, or would it increase GCE suppression of those codons? Because I guess what I'm predicting is that you would get better GCE encoding because you're getting rid of most of the EFR suppression capability. And so now the battle between near cognate suppression and GCE suppression sort of becomes the dominant form. So I'm just kind of asking about that relationship.
Okay, so two questions here. The first is, what the heck is happening during suppression?
Yeah.
So you're right, we get near cognates, and if you look at the genetic code, there's 5 or 6 near cognates for each stop codon. And what we find, we've been doing tandem mass spec with a bunch of different stops, and what we see is a subset of the near cognates being used primarily, and it's usually 2 or 3 for each stop, okay? Interestingly enough, this is context dependent. So of course it's stop codon dependent because there's different near cognates around the different stops, particularly UAA versus UAG, UGA. However, even with the same stop codon, we can get different amino acids primarily inserted based on the local sequence context. And we've mixed and matched the local sequence context from different, regions of stop codons. And we find that both the upstream and the downstream are required. So it's very interesting biology. We don't know exactly how this is working yet, but we're continuing to work on this. And, we're trying to figure out what is specifically happening. Is it the P-site tRNA doing this? Is it the nascent chain in the ribosomal tunnel? There's all these scenarios here. The downstream effects could be mediated by the mRNA that's present in the mRNA channel of the ribosome. So these are things that we have to sort out, and we're working on that currently. Let
me clarify my second question because it was a little sloppy. I guess just to kind of make that a little bit more straightforward, I know that there are some people in your field that are working to suppress, to mitigate these disease effects by adding in suppressor tRNAs.
Yes,
and
like
so
John Lewis.
Yeah, exactly. And so in the context of things like John Lewick's work, adding the small molecules with the tRNAs, do you get like an extra boost, or do you know if anyone's ever tried doing the small molecule tRNA addition simultaneously, or what happens there?
Yeah, so it just turns out that John and I have a multi-PI grant to do this very type of experiments. So we haven't done it yet, but we certainly think, so he might be able to get 60 to 70% efficiency. We think. But the problem is, this is gene therapy when you start putting in tRNAs, and this could potentially be, gene therapy is not easy.
Right.
So what we're hoping is by reducing ERF1 levels while doing the gene therapy, we can get as much or more read-through with much less of the suppressor tRNA. And that's what we're currently testing right now.
Exciting. Thank you so much.
All right.
Very good. And anyone else have any other questions? I can ask one quick question. We have a short second. You had a really quick offhand comment saying that termination is less efficient than read-through. Is that?
No, I bet termination is less efficient than the elongation.
Than elongation, right.
The big peak at the termination codon, right?
Right.
ribosomal
Is there an
profile.
evolutionary reason for that? I guess I didn't really think about termination actually being less efficient than read through of sense codons. But
Well, so think about it. You know, every step of translation is mediated by a tRNA, except for the termination process. That's a protein that actually has to recognize a stop codon. That's unheard of at any other stage of translation.
Right,
And in
right.
fact, it's been shown that the first base after the stop codon is actually pulled into the A site, and it's involved in recognition. So it's actually a tetranucleotide termination signal.
Hmm.
And so I think the difference here is that you've got a protein recognizing instead of, you know, tRNA codon anti-codon interactions, right?
Right, right. No, that makes sense. Very good. Great. All right. I think we're thanks again, Dr. Bedwell. Fantastic talk. And I think we'll move on to Philip.
All right. I'll
All right,
stop
thanks again.
sharing.